Hello everyone, and thank you for listening to my project and this presentation. So my name is Margarita Damai, and I'm a second-year student for biomedical sciences here at London Metropolitan University. And today I'm here to talk about my project, that is the synthesis of novel pyrrolo quinoxalines probe for Cooper detection and its use onto the healthcare, environment, and pharmaceutical field. So today I'm going to talk about the relevance of Cooper, how we were able to synthesize our fluorescent probes, and then the studies that we performed, which were the fluorescent, NMR, and binding studies. And most importantly, we are going to discuss how our probe can be applied across different fields. So to begin with, why Cooper? Why it concerns us? Well, Cooper is a naturally occurring metal that has further applications, not only in the environment, but also within our organism. For example, in combination with proteins, it can be used to produce enzymes, and as we know, enzymes catalyze various bodily reactions and functions, without which we won't be able to survive. So this inevitably means that Cooper's misregulation can link to different diseases such as Alzheimer's, Wilson's, Parkinson, and even obesity or diabetes. Therefore, throughout the years, various methods have been developed to detect Cooper levels, such as, for example, ASV, ASS. All these methods are very expensive, though. They require a lot of time and a lot of trained staff to perform them, which makes them not always very handy. Throughout the years, though, fluorescent spectroscopy has been an alternative that seems to be less time-consuming, less expensive, and does not require a lot of trained staff. And here is where our project comes into life. We wanted to create a fluorescent probe that would be able to specifically detect Cooper. So based on the literature result, pyrrolo quinoxalines scaffolds seem to be very potent. And its analogs have been considered to have fluorescent activity, such as, for example, for substituted pyrrolo quinoxalines hydrogens, which have been effective for amyloid fibrillins. Therefore, we aimed at creating a substituted pyridine pyrrolo quinoxalines, which we will call this PQ or probe 1, that would be able to specifically detect Cooper, and this would be attributed due to the nitrogens into its structure. So in other words, the aim of our project was to create PQ probe 1, check that it is selective and specific for Cooper, and further test its application onto the field. So to begin with, to synthesize our PQ, so probe 1, we performed a three-step reaction, which started with a nucleophilic aromatic substitution reaction, followed by a reduction, and in the end by cyclization through using a modified Pictet-Spengler reaction that resulted into our PQ into good yield. So after we synthesize our PQ, we wanted to check that it is selective and specific for Cooper, so we decided to perform different studies involving fluorescent, NMR, and binding studies. So to begin with fluorescent studies, we wanted to check that PQ can specifically bind to Cooper and be selective to that. So this would mean that the fluorescence of PQ together with Cooper would decrease. So when we measure the fluorescence of the PQ alone, you can see that there is a high intensity, whereas when Cooper was added, the intensity decreased. So basically, this means that PQ and Cooper quench the fluorescence, and means that they specifically work with each other. But we wanted to check that this is selective only for Cooper. So what we did, and this is in figure 2b, was that we used different metal salt solutions. We treated them with PQ, and we measured their fluorescence, which is in blue. And as you can see, there is a high fluorescence. But then we decided to add Cooper within them. So once Cooper was added, you can see that the fluorescence dropped. This meant that the presence of Cooper within those metal salt solutions and PQ would decrease the fluorescence, which meant that PQ is specific and selective only for Cooper and not for any of those metals that were used. Furthermore, we decided to perform NMR studies. This would mean that if Cooper interacts with PQ, due to Cooper being paramagnetic, the peaks would shift. right? So as you can see onto the diagram and to the figure 3, there are 3 diagrams. So diagram A is just the hydrogen and NMR spectra for the PQ, and in B and C we have the presence of Cooper. So in B we have a 0.5 equivalence of 1 to Cooper, of our probe 1 to Cooper, and into the C we have a 1-to-1 equivalence of the PQ and Cooper. So as you can see, the peaks start to become broader once the equivalence is 1-to-1. And finally, we performed binding studies. This was to check how the interaction and the nature of interaction between PQ and Cooper would be. So we decided to break these binding studies into 3 parts: into Cooper detection limit, into EDTA decomplexation, and into the binding stoichiometry through a Dobbs plot. So initially we wanted to check what the detection limit for Cooper would be. So what we did was that we treated our PQ with different concentration of Cooper, starting from 0 to 1,000. And as you can see, the fluorescence started to drop at 250, meaning that the detection limit starts from there. So as you can see onto the image here, as the concentration of Cooper increases, so from left to the right, the fluorescence starts to decrease. As we mentioned, we wanted to check the decomplexation. So into figure 5, you can see that we have measured the fluorescence of Cooper, of PQ alone, of PQ together with Cooper, and PQ, Cooper, and EDTA. So if EDTA would reverse, basically in theory, EDTA can destroy the complex of the PQ and Cooper and increase the fluorescence again. So to prove this, basically we measure the fluorescence of PQ in blue, and as you can see it is high, and then we measure the fluorescence of PQ together with Cooper, which should in theory be low, and that's what it was, because the fluorescence would drop because they interact with each other and quench the fluorescence. So if EDTA was to be added and in perfect condition, if EDTA would break the complex, then the fluorescence will increase. And as you can see onto green, the fluorescence increased, which meant that this reaction, the reaction between PQ and Cooper, is reversible, which means that our probe can be recycled, which is positive because we can use it for again and again into the same reactions. And finally, we used the Dobbs plot to check the binding stoichiometry, which was a 1-to-1 ratio between PQ and Cooper, as is shown into figure number 6 through the Dobbs plot. So far we synthesized our probe and we checked through different studies on its nature of interaction of the PQ together with Cooper. But now we want to check its application. And for this, we are specifically talking about the environmental application and bioimaging application. So to begin with, in terms of environmental one, we used our probe for water analysis because our probe can be used to detect Cooper level within the water. According to the regulations as of 2016, the concentration of Cooper within the water sample is 2 ppm. So what we did was that we collected water samples from different sources, and we wanted to check whether or not the levels of Cooper within those sources would be the same as the level of Cooper that has already been detected by other methods such as MBEAS. So basically we wanted to compare the reliability of our probe with a current well-known and well-used method. So after we did our measurements, we came into the conclusion that the levels of Cooper that our PQ detected was the same level as the one from MBAES. So this meant that our probe, which is less expensive, which is less require less time and less specialized staff, can produce the same reliable result as a very advanced, expensive, and time-consuming method. And to round up into biomedical field, we decided to check for cell imaging. So basically our probe can be applied for fluorescent imaging of Cooper within the living cells. So what we did was that we treated cells with PQ alone, and you can see there's fluorescence, and then we treated them with Cooper at a concentration of 400 micromolar. And as you can see into figure 8b, the fluorescence completely dropped, which meant that our PQ interacts with Cooper, and this allows us for cell visualization. So in conclusion, this project presented a way of how we were able to synthesize a novel pyrrolo quinoxaline fluorescent probe that is specific, selective for Cooper. Basically, it can be applied into the environment for detection of Cooper level within water, and bioimaging to produce and check traces level of Cooper within the organism. So I want to give a special thank you to my two supervisors, Dr. Bhavan Patel and Dr. Priyanka Ganguly, for their support and for their patience in supporting me throughout this journey, because it is really challenging sometimes to work with undergraduate students. So I really appreciate their hard work, and I think that they deserve a recognition for this. So I have also put our contact information into here in case you would wish to ask us any question or to get in touch with us or has any proposals. So feel free to contact either me or my two supervisors. And I really want to say thank you for listening. And if you would have any questions, please feel free to send us an email, and we would be more than happy to